Okay. So the last speaker of today is Celia Barroux. She's a PI in the University of Zurich, and she's going to show us a non-random spatial distribution of the transcriptional compartment in Arabiopsis italiana. So thank you very much, Celia, and leave it with you. I'll join you later for the questions.
Thank you very much, Sarah. I just need control on my presentation, which I don't have now.
Sorry, yeah, I can do that for you, no worries. Sorry, I forgot, you're right. My presenter, so now it's working now?
Yeah.
Okay, perfect.
Very good, yeah. So good afternoon, everyone. It's my pleasure to present you some of the recent work we have been doing. All of this is unpublished data, and we still have a long way to go to gather data for publication, but I think it's nice to share it with you at the moment. So we are interested in the transcriptional compartment, but first let me put you in context. We are fascinated together with other people with the fact that some transcriptional changes can be extremely rapid upon environmental cues in plants. I'm giving here for you two examples where Arabiopsis transcriptome is completely rewired under pathogen infection, here by FLAG22 treatment or due to light cues. And you can appreciate the time in minutes on the top of this heat map of gene expression, which shows you how massive and rapid gene expression changes can be. So in this context, we are wondering how it correlates with the spatial organization of transcription in Arabiopsis nuclei. What is known already is that for some genes, there is a dynamic spatial repositioning happening upon transcriptional activation, and there are only a few cases which have been described and documented. One case is of the CA gene cluster responsive to copper in the medium, which is relocated during activation, transcriptional activation. So relocation happens slightly after transcriptional activation and reinforces activation. That means if you interrupt relocation, you do not lose activation, but you lose the strength of transcriptional activation. And another example is of the CAB locus, which becomes repositioned towards the periphery upon light. So it's a light-induced gene locus. So the question is whether gene repositioning is a local event for only a few genes that are really highly responsive to environmental cues, or is it a general rule when such massive transcriptional changes are happening? And actually, we realize that we don't know many things about the transcriptional compartment in the nucleus. Is it randomly distributed, or does it follow a certain functional spatial pattern? And I'm just highlighting for you below some examples of patterns that it could follow. Okay, so transcription is known already to not be homogeneously distributed, and in animals there is a concept of transcription factories, which describes the idea that transcription is actually that transcription, yes, can be visualized as discrete foci, which are visible here as a BR UTP labeled RNA, or on the right side with immunostaining of some active isoform of RNA Pol II, and in animals, these transcription foci have a lifetime of about 5 seconds. Okay, I think the version which is uploaded now is a former version. It's not the newest one. I had a very nice image of a state image, but it's okay. So organization is not in the organization in discrete transcription factories is not prevalent in plants. We see rather reticulated pattern in Arabiopsis, and in wheat, however, we can see foci which could be associated with the notion of transcription factory. And in Arabiopsis, what we also know is that RNA Pol II forms cluster of variable size. This has been described using single molecule imaging done by Schubert. And he described small clusters or large clusters containing different number of molecules, and that he could measure the appropriate diameter. Okay, now to analyze the spatial distribution of the transcription compartment, we have developed a workflow. we isolate nuclei from leaves, embed them in a acrylamide pad, and we immunostain them for RNA polymerase II and DNA using state imaging, and then we perform 3D segmentation using IMARIS. We then segment actually the RNA Pol II clusters of variable size using a growing spot algorithm in IMARIS. And then we export statistics associated with the RNA polymerase II clusters and in terms of distance, shape, volume, intensities. And we feed this in the new software that we have developed that allows for interactive data analysis. We also, this software can handle several levels of data, including genotypes, treatment, and image replication. So now about the biological systems that we use to compare whether there's a dynamic change in transcription. We use the so-called dark to light transition, which Freddie Barnage Labs is actively working on. And we know that during the dark to light transition, which is happening after termination, there's a massive transcriptional reprogramming, also chromatin reorganization at the nuclear scale described by Clara some years ago, and also RNA Pol II activation that you can measure quantitatively, shown here on the right side. So that's our system, that's our approach, and now we have a nice collection of 11 nuclei coming from shortcut sets, so dark samples and 11 nuclei from light samples, and we have segmented all of them. Now I'm just showing you an easy representation of RNA Pol II clusters, which are just classified in two categories for high intensity and low intensity. And already on this image, you can appreciate that there seems to be kind of a radial distribution of RNA Pol II with a lower intensity of RNA Pol II cluster at the periphery. So it's wrong, it's not depleted, but it's a lower intensity at the periphery. And it seems to be redistributed in the light sample. Now, when we look at it more quantitatively using DataVis, we observe two things. First is that the cluster themselves, so the way the cluster are distributed, is changing between samples, dark and light samples. And as you can appreciate here on the graph here, showing density distribution corresponding to the distance to 9 nearest neighbor of the RNA Pol II cluster, there's a strong relaxation of RNA polymerase II distribution in the nuclear space. At the same time, there is definitely activation going on, and you can read this in this scatter plot showing you a volcano eruption of RNA Pol II intensity, and now this is intensity of RNA Pol II, which is normalized over intensity of the chromatin. However, don't be misled by this volcano picture, because if we actually count the number of data points in this graph, it's only the highest intensity RNA Pol II cluster represent less than 1% of the total transcription compartment. But it's significant and it's real. Now, when we also look at some RNA Pol II cluster in relation to their distance to the nuclear periphery, so to back up the image I've showed you before, you can also appreciate, so here on the x-axis is the nucleus to the nucleus periphery, distance to the nucleus periphery, 0 being the boundary, as we define in our segmentation, and the minus numbers correspond to the center of mass of the nucleus. And on the y-axis, you have, again, this normalized intensity of RNA Pol II. And you can see that activation of RNA Pol II occurs primarily for clusters which are towards the periphery of the nucleus. So they are more engaged in this activation. And last observation for you today is that the RNA Pol II clusters, they change in their chromatin compaction. If you now look at the signal density of chromatin inside the RNA Pol II cluster, you can see that the signal density distribution is changing in the light compared to the dark treated samples, and we can interpret it with this schematic way that chromatin gets relaxed within RNA Pol II clusters when in samples, so-called light samples. So with this, I would like to leave you with two levels of conclusion. One is more scientific and the other one more methodological. We show that there is a reduced transcriptional activity at the nuclear periphery, and that might be consistent with repressive domains that have been described before, so LADs and also heterochromatin, which is localized mostly at the periphery. RNA Pol II cluster landscape is also quite dynamic. We have seen it in forms of spatial relaxation. There's also a densification of RNA Pol II per cluster, reduction of packing density in samples that have been exposed to light. Lastly, transcriptional activation seems to follow a radial gradient with increased participation of cluster at the nuclear periphery. Here I would like to put a note of caution. Nuclear periphery, in our case, it's in a large sense because we don't have a nuclear membrane marker. And as you could see from the graph, I mean, it's happening within 500 nanometers, so it's far away from the membrane. And methodologically, we have also now established a workflow from sample preparation to data, which works very well to do this spatial analysis. And we also have developed an interactive visualization interface for data mining because these are a huge number of data. And this is publicly available. I will give you the address. I would like to finish with the acknowledgements. The work was initiated and largely experimentally done by Ricardo Ronda, a postdoc in my group, who left now, and I also completed the data analysis, image analysis. Devin Ross is developing dataviz for us, and we have nice collaboration within the InDepth network with Barnish Lab and to some extent Daniel Schubert's lab, but I haven't shown you any data here. And that's it. I just leave you with these usual links that you want to consult, perhaps. It's spare time. Thank you for your attention.
Thank you very much, Celia, to show us, for showing us so nice pictures and this interesting topic. So I'm going to check if there are any questions. Otherwise, I will go ahead with my question. Okay, so what do you think that is what is keeping the RNA polymerase out of the periphery, of the nuclear periphery? Is the compactation of the chromatin there, or what do you think?
Yes. So actually, there are two things. One, what I wrote in my titles, they are depleted. In a way, we also see that it's depleted when we do density distribution of the RNA Pol II across, compared to the membrane, to the boundaries, and we see there are less foci, but it could also be due to the fact that you have chromocenters that exclude them, so the LADs as well. You still have RNA Pol II foci towards the periphery, but there is a broad band below the nuclear periphery where the RNA Pol II is less active. So it's not only the numbers, but also the clusters themselves are less active. And then what's the mechanism for this? I don't know, but it fits very well with the idea that you have anyway repressive domains there. So that's a bit of the chicken and the egg question. Yeah. Okay, great. Just checking again, since the questions are not coming yet. You have focused on dark and light. Mm-hmm. To analyze this reorganization of the nucleus. But can we expect that this is for any kind of stress or environmental change, or do you think that it's going to slightly depend on the environmental cue that is the plant is perceiving? This I don't know, and that will be the next question. Of course, at least now what we know what we are looking for, because before we had no idea whether we would have at all a spatial dynamics of the transcriptional compartment. It could have well been that the distribution remains the same, and you just have more processing of Pol II on the same site. Now we know that we can look indeed for a spatial shift and for a different clustering effect or distance between clusters, and we can look now at other stress conditions.
Okay, so there are no. Here is a question coming. So thank you for the presentation. This is Ahmed Tech, and he's asking, do you expect similar observations with other RNA polymerase enzymes? Not just two?
Also polymerase isoforms, you mean? Perhaps. Or I just didn't hear very well.
Perhaps yeah, well, the question is other RNA polymerase enzymes.
Pol 4, Pol 5. No.
Yeah, that way I understood.
We don't know. We just started. No, so I can't tell you anything. And no, we need to immunolabel. We could immunolabel other RNA Pol 2 isoform, Pol 3, Pol 4, but no idea. No, it's very fresh work.
And anyway, the one that you are, following is the the one phosphorylated in serine 2. That is
Serine 2, yeah.
That is the one that it goes elongation
Yeah,
phase.
it
Is that
characterizes
correct?
primarily the elongation process, exactly. So at least we are targeting the active compartment of RNA polymerase II. We have already some data for serine, for RNA polymerase II phosphorylated in serine 5, which is supposed to be more in the engagement. And then we seem to see a difference. That means there might be a difference between isoform in their behavior, but we need to produce more images for that.
Okay, so great. I think that there are no more questions coming. Okay, and anyway, we are 10 minutes past the time for this session, so I think that we should close here. And thank you very much again, Celia, and all the other speakers and everyone that was attending today's session, the previous one and this one. So thanks a lot for all the contributions and the nice discussion. Thanks, Clara, also.
Thank you, Sarah.
Okay,
So,
thank you very much.
Bye, everyone.
Bye-bye. Bye, everyone.